Thank you. This is being recorded, and for those of you who are no doubt going to ask, the slides will be available and the webinar recording will be available probably next week. Although I hope most of you who are based in the US will enjoy Thanksgiving holiday and not be on trying to find these slides and this work. We promise we will send you, every registrant, an email with a link to it, as well as putting the link on our MRCT Center newsletter after this webinar, so you'll have access to it. My name is Barbara Beerer. I'm the faculty director of the MRCT Center, the Multi-Regional Clinical Trial Center. And it's really my distinct pleasure to welcome back to the stage, as it were, virtual stage, Daniele Bertolini, to do the second round of a presentation on the use of AI digital twins and synthetic data. For those of you that missed his prior discussion, I'm going to bring you towards that a little bit later, but I do think it's worth going back and listening to the podcast or the webinar recording and seeing the slides, because it is probably the best introduction I've seen. So with that, let me begin and welcome all of you. Where today we're going to be unpacking practical use cases for clinical research, discussing digital twins, and ending with a few comments on synthetic data. Could I have the next slide, please? This is being recorded for you all. And I just want to say, as a disclaimer, views expressed today are not endorsed or reflect the views of Mass General Brigham, Harvard Medical School, or any other affiliated or unaffiliated organization or entity. Our center is supported by voluntary contributions, which are reflected on the website, as well as by grants, and we are committed to autonomy in our research and to transparency. Happy to discuss that further. Could I have the next slide, please? So thank you so much for joining. Today, we are in a webinar session, so please put your questions as they come up in the Q&A. We'll do our best to reserve some time at the end to answer live. And don't put them, if you could, into the chat, because it's very, very hard to follow them for us. If you do that, we will also make every attempt to answer them later if we can't answer them in real time. You can use closed captioning, which is on your Zoom link under CC Show Captions, if you would like to use closed caption enabled recording. Most of these links in our presentations will be shared in the chat so that you'll be able to pull them and get them. The meeting is being recorded, as I mentioned, and we'll be making these available next week. Do sign up for our newsletter, which gives you all of the things that we're doing on the MRCT Center website. Could I have the next slide? So the MRCT Center, for those of you who are not familiar with it, is the Multi-Regional Clinical Trial Center. And we are a research and policy center focused on addressing the conduct, oversight, ethics, and regulatory environment of international clinical trials, with the ultimate aim to improve the integrity, safety, and rigor of global clinical trials. And we do that work by identifying emergent or never solved issues in a clinical research ecosystem and then do more than just define them or write a white paper about them. What we try to do is to develop ethical, actionable, and practical solutions or approaches. And we do that by pulling together people who know and are involved in the question that we're asking or trying to do something about. So it is my distinct pleasure to say that when we came upon this issue of digital twins and synthetic data, we turned to Daniele for the expertise that he could share here. Next slide, please. So you will be hearing from me, and then, as I said, from Daniele Bertolini, who is the managing machine learning scientist at Unlearn.ai. Could I have the next slide, please? Now, when I started this, we defined digital twins as referring to simulated models of individual patients designed to predict disease trajectories and/or treatment responses, which offer the potential to enhance statistical power, optimize the trial design, and/or limit the number of participants assigned to a control arm. While synthetic data, so this is all based on an individual and then predicting their outcome by sort of modeling that through the kinds of data that we have available in terms of trajectory, disease understanding, and other comorbidities and cofactors. Synthetic data, on the other hand, are artificially generated data sets that mirror the statistical properties of verified clinical trials. Both technologies are being used for the design, conduct, and analysis of clinical trials, and both with the intention of use for regulatory submissions, although in very, very different ways. Could I have the next slide? So when I first started down this path, I had no idea what a digital twin was. I sort of thought it was looking at myself in the mirror, and then found that, it was worth unpacking the concept of digital twins. Could I have the next slide? We therefore invited Daniele to give us a sense of where and how digital twins and synthetic data are applied and used in clinical trials, where they come from, what their ability and constraints are, and how they could be used. Could I have the next slide, please? So at that point, which was about a month ago, he took us through three basic themes. One is, how could the digital twins be used and what they were and how they're useful, how they're built so that we had a conceptual understanding of how they are derived, but not a technical understanding that is far beyond our capabilities, and then how to use digital twins. You'll see in the QR code the link to the website, which is on the right, which gives you the link to that webinar and slides. And I must say, it's the best one that I have heard. Could I have the next slide, please? Now, you'll see there when you link to it that you can view the webinar. You can also download the slides. Personally, I think most people go and download the slides, but it is well worth spending the time listening to the webinar, because it really allows this to come into a full appreciation in a way that just looking at the slides does not. Daniele is really a master educator, and therefore we invited him back to talk to us about various use cases in the use of digital twins and then with synthetic data. Could I have the next slide, please? And with that, I would like to turn it over to you, and thank you once again for spending the time with us and helping our education and understanding of this complex area. Thank you.
Thank you, Barbara, for the very kind introduction. And also thank you, everyone who decided to join us today for this second second episode of the Digital Twins webinar series. As Barbara mentioned, today I will be discussing applications of digital twins and synthetic data to clinical trials. Next slide, please. So this is the agenda. I will first just go through a quick recap of some of the key concepts that we have discussed in the last webinar. And in particular, I'll refresh the key ideas behind digital twins and synthetic data, which are kind of foundational for this discussion. And then I will spend most of my time discussing some specific applications of digital twins and synthetic data. In particular, I will discuss 5 of these applications. I'll discuss what's called Procoa and its Bayesian version, which is a use of digital twins to boost power or reduce sample size in randomized clinical trials. I'll discuss how digital twins can be used to construct synthetic controls in non-randomized trials. I'll briefly discuss how digital twins can be used to discover responder subgroups. And finally, I'll close with an example of how synthetic data, kind of a broader concept, can be used to optimize the design of clinical trials. So of course, I will not get into the details of any of these applications, but I'm hoping to, kind of convey the key ideas behind it. And most of all, I'm hoping to convey the breadth of applications that this technology can enable. And of course, those are just examples, but I hope they'll be helpful and inspiring to people who are listening. Next slide, please. Okay, so let's start with just refreshing what is a digital twin. Barbara already did this, but for the purpose of this discussion, a digital twin of a patient is a computational model of that patient, of that individual patient, that makes a prediction for future outcomes of that particular patient. And the idea is that you take, for example, a slice in time of the characteristics of a given patient, let's say at the beginning of a trial, for example, that is the input to a digital twin generator model. And what the model gives in output is the digital twin of that particular patient, which is a probabilistic prediction for how the different outcomes will evolve over time, typically under standard of care or placebo. Next slide. And what you see here is, just a concrete example of what a digital twin might look like. So in this case, we are considering a patient who has ALS, and you will see at the top of this table, there are some demographics and medical history information about that patient. And in the section below, you see a table. Each row is a different variable, a different outcome that I'm measuring on this particular patient. Because this is a patient with ALS, you will notice here, some lab tests and some particular scores that are used to track the progression of ALS. And the columns here, you can think of the columns as being time. So the white column is baseline. So those are the value of each one of these particular variables for this particular patient, let's say at time 0, at the beginning of the trial. So those are observed values. Those are, along with the static kind of a demographics and medical history variables at the top of the table, those are the input to the digital twin model. And what you get back in output is the purple columns, is how this particular patient, given its baseline at a particular time, how this particular patient will evolve over time. And so you can think of column 1 and 2, for example, as visit 1 in a trial, visit 2. So it could be 3 months after baseline, 6 months after baseline. And the values that you observe there, those are the predicted values of these different variables. And this essentially constitutes, in this particular example, a digital twin of this particular patient. One thing you will notice, which we'll get back to it later, is that the predictions are not just numbers. Like this particular patient, albumin at baseline was 42. Let's say after 3 months, so at visit 1, the prediction of the model is not just 42.2. You will notice that there is an error bar there. And that's because the underlying model, it is in fact a probabilistic model. So what the model is learning from the data that is trained on and what it's trying to predict is not just a single point prediction, but kind of a probabilistic distribution over the possible outcomes for this particular patient. So you can think of this as a way to quantify to not just provide a prediction, but also to quantify the uncertainty associated with that prediction. Next slide, please. One thing I wanted to clarify before we move to applications that Barbara already did very well, but I kind of want to stress it again, is the difference between digital twins and synthetic data. I would say that there is no universal, accepted, clear book definition of what exactly is a digital twin and what exactly is synthetic data. But for the purpose of this webinar, or really for the purpose of this series, what I mean by digital twin is essentially what I just described. You take the input from a particular patient and then you predict the progression for that patient over time. What I mean by synthetic subjects, which for simplicity I'll refer to as sort of a synthetic data, is something similar, but in this case, there is no input from any particular patient. Synthetic subjects are hypothetical patients, and their hypothetical progression, but those are no real patients. Those are just hypothetical cohorts of patients that will have similar statistical properties as true as real patients, but they're not associated with any particular real world patient. A simple example of this is just you can think of the idea of defining some inclusion-exclusion criteria for a trial, and then generating patients that satisfy these synthetic subjects that would satisfy these criteria, and use those to simulate a future trial. And one thing that we discussed in the last webinar is that if you have a expressive and sophisticated enough machine learning model that learns disease progression from historical data, then you can use, in fact, the same model to either generate digital twins of patients once you input specific characteristics of a patient, or you can use the same model to generate synthetic subjects. And in this presentation, in the rest of the presentation, I'll discuss applications of both digital twins or synthetic data. Next slide, please. Okay, so let's dive into some of these applications. What I have here is kind of a schematic, kind of a high-level representation of a timeline for drug development that goes through the standard phases. We go from phase 1, where we typically want to establish whether the experimental treatment is safe. Moving on to phase 2, we want to know if the treatment is effective. Pivotal phase 3, we want to understand if the treatment is better than the standard of care. That's typically when we run large randomized trials. And then phase 4, that has to do with post-approval information that I need to gather about the treatment. For example, what is the long-term effect of the treatment? What is the long-term safety of the treatment? How does the treatment work on real-world populations, and so on? And in the bottom part of this timeline, I kind of put examples of where and how you might use digital twins or synthetic data to improve evidence generation at each one of these steps. And improving evidence generation means also de-risking the outcomes of this whole process, which is long and expensive typically. So for example, after you run a phase 1, you could use digital twins to help detect early efficacy signals, even though phase 1 is not typically for efficacy, but you might want to try to understand very early on some early signals of efficacy. And you can use sort of synthetic data to optimize the design, using the information that you gather in the phase 1 to optimize the design of the phase 2, or even optimize the design of a combination of phase 2, phase 3. Then you run your phase 2. There you can use your digital twins to, now your goal is to really kind of understand if the treatment is effective. Often, the sample size of phase 2 trials is not very large. And so it might be, in some cases, difficult to detect efficacy. So that's another place where you can use digital twins to boost the power, so to boost your chances of detecting an efficacy signal if, in fact, the drug is effective. You can use digital twins to detect some responder subgroups, or more broadly, optimize the design of the next phase. In fact, you can use digital twins to design a phase 3, which is smaller, faster, and cheaper. Then you run your phase 3, and again, you can use this tool, these digital twins, to boost the chances of detecting efficacy, to again detect relevant subgroups. And then finally, when you run your phase 4, if the type of studies you're running are non-randomized, for example, open label studies, then you could use digital twins as a sort of a synthetic control to compare your treated patients to. So this kind of hopefully gives you an idea of how digital twins serve and synthetic data can help in different ways at many different stages of the drug development lifecycle. Next slide, please. So then if I take all these different applications, I can kind of group them into 3 main categories. So one category has to do with optimizing the design of a trial, which of course I can do it for phase 2, phase 3, or phase 4. One big bucket has to do with improving the analysis of a trial that you have run. So you could boost the power, or you could reduce the sample size. If you're running a non-randomized study, you can use a synthetic control to analyze your results, or you could use it to redefine better endpoints and subgroups. And finally, there are different categories that has more to do with phase 4 type of studies, where again, you might use external controls to answer some post-approval market access questions. So what I'm going to do now is discuss essentially briefly, but discuss all of these applications that I put into these 3 buckets. Next slide, please. All right. So here is the first application. So we have discussed this a little bit last time, but I'm going to refresh the idea here. So the idea here is to use digital twins to boost the power of a trial, or alternatively, to prospectively reduce the sample size needed to achieve the same target power. So this application is called Procoa, which stands for prognostic covariate adjustment. And here is how it works. So covariate adjustment is a standard technique that's often used in the analysis of a trial. And the idea is that you can include certain baseline covariates in the analysis of your outcomes. For example, age or sex, let's say. And if those covariates are correlated with the outcome that you are analyzing, then including these covariates in the analysis of the trial will let you explain away some of the variance in the outcome. And because you can explain away some of the variance of the outcome, because some of that variance is due to unbalances between the two arms in sex, age, or whatever other variable, because you can explain away some of that variance, the result is that when you go and try to estimate the treatment effect, let's say for a continuous outcome, just as a sort of a difference in means between the treated and controls, because you're including these variables and you're explaining away some of the variance, you get a more precise estimate of the treatment effect. So you get a point estimate, you get an error around it, and that error is going to be smaller. And that smaller error allows you to do many different things because that error is related to, on the one hand, is related to the power of the trial, so your probability of detecting the effect. The smaller the error, the more precise the estimate, the more powerful the trial is, so the more likely it is that you can detect an effect if there is an effect. Alternatively, if you want to keep the same target power, then you need fewer subjects because you have a more precise estimate of the treatment effect. Now, this is a standard mechanism. You can include age, sex, or different other baseline covariates. The idea of Procoa is that all you need to do is to include one single variable, which is the digital twin prediction of the outcome for each individual patient. And not only this is sort of using a single variable instead of many potential variables, but because the digital twin is the prediction of the outcome for that particular patient, you can actually mathematically prove it. If you have a good prediction for it, this is the best that you could do. It's the best adjustment that you could possibly do. Of course, if you have a good model that's able, that's capturing the dependence on all the different baseline covariates, and it's making a good prediction for the outcome. So you see that what's happening here, we're making use of historical data of past clinical trials to train a model, generator model that's learning from that past historical data. And then we're using that same model to make a prediction for the future control outcome of each patient in the trial. And we're using that as a covariate. Instead of adjusting for age, sex, we're adjusting for the prediction of the outcome for each individual patient. So the way the past information, digital twins entered through in this analysis is not by adding new participants. This is just a standard RCT. It's via a covariate that you're adding to your analysis that in itself, it was built using some information about past clinical trials. Next slide, please. So, all right, so this is kind of just a schematic representation of how this might work. You enroll a patient, the patient get randomized in a standard simple RCT, either to the active arm or the control arm. The baseline data prior to randomization enters as an input the AI model. The AI model creates a digital twins for each patient in the trial, either in the active or control arm, and that's going to be used in the final analysis in this sort of a covariate adjusted fashion that I just described. Next slide, please. So, okay, we have already discussed this, but essentially once I have this covariate adjusted analysis that gives me a more precise estimate of the treatment effect, now I can use the fact that I have the expectation that I have a more precise estimate of the treatment effect, essentially in two ways. One thing that I can do is that I could keep the same target power as my standard design of the trial, but because I expect a more precise estimate of the treatment effect, I need fewer patients. So I can run the same trial with fewer, in particular, control patients. I could reduce the overall size or the control arm size. Probably I want to reduce the control arm size. Or vice versa, I want to keep the same size, but I can spend my expected smaller uncertainty on the treatment effect essentially as a power add, so I can boost the power of my trial. Next slide, please. Okay, so this is all great. You might ask, does this work? What is the actual benefit? Show me some numbers. Okay, so I'll show you some numbers. So here is a collection of past clinical trials that you can see they are in either Alzheimer's disease, ALS, or Huntington disease. Those are trials that are either in our historical data set or they've been reanalyzed as part of a collaborations that we have had with different pharma companies. And so what we have done here, so here are some stats about this, these trials. You can see the different sizes, different phases, durations, endpoints, etc. So what we've done here is to take each one of these trials and reanalyze it so we generated digital twins for each patient in this trial, and we have reanalyzed them and then compared and asked the question, for example, how much smaller of a trial you could have run had you used digital twins in this particular trial, or how much power you could have achieved if you had kept the same sample size, but you had used digital twins in your analysis. And here are some numbers. Next slide, please. So the columns here are the trials that I just described. And first look at the middle column, the Procoa variance reduction. So this is the reduction in uncertainty of the treatment effect that I mentioned. So this is if you had used digital twins in each one of these trials, you would have reduced the variance on the treatment effect, so the variability of your treatment effect estimate, anywhere between 10 and 20%. That is a large number, especially if you compare it to the left column, which is the amount of variance reduction, so increase in precision that you would have got had you adjusted just for the standard baseline covariate in that particular trial. So you see that those are much smaller numbers and much more variable across the trials. Okay, now you could ask, great, I have this variance reduction. What do I do with it? Well, the right column now is telling you a much more practical number. This is telling you if I had used digital twins prospectively in each one of these trials, given this increase in precision, I could have run the same trial with the same target power, but with a much smaller control arm. And you can see here the fractional reduction that you could have got, which goes from anywhere from 20 to 30%. And of course, smaller control arm means reduced cost. It is very expensive to enroll patients in a trial. It means reduced time to read out because it takes a long time in some of these indications to enroll patients. And so it means shorter time to, the outcome of the trial. If the drug works, it means shorter time for the drug to get to patients. Next slide, please. So one thing I wanted to mention is that we already mentioned in the previous webinar, but this particular application, so using digital twins as covariate in the analysis of a continuous outcome, has been qualified in different ways by regulatory agencies. So we went through a qualification process with the EMA, and you can, this was back in 2022. You can, Google EMA Procoa qualification. You will find the entire qualification, but here there is just a, extract from it. The FDA does not have an analog kind of a qualification process for the method itself. So it's more on a case-by-case basis. However, here I'm reporting the response that the FDA gave us when we applied to the I-STAND program, which essentially says that they concur with the EMA that Procoa is effectively a special case of covariate adjustment, and so it does not deviate from the general covariate adjustment guidance. And then, of course, each case needs to be reviewed on a sort of a case-by-case basis. But I will mention that we have had interactions with FDA on some of this, particular programs with positive feedback. Next slide, please. All right, so this is one question that was brought up at some point, which is, Procoa uses historical data in some way. We have already described essentially how historical data is used. Historical data is used, so past clinical trials or observational studies are used to train a model that learns patterns from these large data sets. And then once the model has been trained, can be used in a new RCT to generate these digital twins, which can be used, for example, as covariate in this Procoa application. And so the analysis itself is a standard pure frequentist analysis. As such, controls as strict control of type 1 error, so can be used for phase 3 type of analysis. Now you might ask, there are other ways to incorporate information about historical data into the design and the analysis of an RCT, such as Bayesian methods. So those also can improve efficiency. But a Bayesian analysis is, it's sort of conceptually and fundamentally a different statistical framework to do inference. In this case, you use historical data to set some prior distribution on the parameters of interest. For example, you might set a prior on the expected average control response based on historical data. And then your prior distributions get updated with the observations in your actual trial. Typically, Bayesian analysis do not guarantee strict control of type 1 error, but they can give you much larger gains in efficiency. So these two approaches, they can actually be combined. You can actually do both. You can do a Bayesian analysis that includes digital twins as prognostic score in a covariate adjusted fashion. So you can kind of leverage the advantages of both Bayesian analysis and Procoa. That's what we call the Bayesian Procoa. If you go to the next slide, please. So here is an example. I'm taking the same equation that I had before that just simply represents a kind of a linear regression model that you might use to estimate treatment effects when you have continuous outcomes. And now the parameters here that enter in this regression, they become themselves random variables, and you can set a prior distribution on it. I will not get into the details, the technical details of this, but the key idea is that you can combine advantages of Bayesian analysis with advantages of Procoa. The thing you have to keep in mind is that, as in any Bayesian analysis, you don't have strict control of type 1 error. So this type of analysis might be more suited for earlier phase analysis, maybe a phase 2 analysis. So next slide, please. What you see here now is, this is the same studies that I presented before, where we have done a reanalysis earlier we did a reanalysis using Procoa. Those are the Procoa columns here, the second and fourth. Now we have reanalyzed this same study, but using Bayesian Procoa. And so same idea. Now, if you focus on the last two columns, the next to last is the control arm reduction that you could have got using Procoa. We already discussed earlier. Now you see the jump in performance here. That's because Bayesian Procoa can give you an even larger boost in precision of the treatment effect, which translates into in this example, an even larger reduction in the control arm, or even though I didn't report it here, you can translate that into a boost in power. And you can see now that in this case, the control reduction goes, it moves from being between 20 and 30 to being between 30 and 40%. Next slide, please. Okay, so I'm going to jump now to a different application. So far we talked about RCTs and how digital twins can be used to boost power, reduce sample size. What if you have a single arm trial? So of course, randomized control trials are the gold standard to establish efficacy of some experimental treatment. However, in some cases, for example, where randomization might be unethical, for the case of rare diseases, for the case of pediatric trials, in early phases trials, in some cases for accelerated approval, especially for oncology trial. There are some situations where a single arm trial where everyone gets the treatment and there is no concurrent control might be used. And so when you have such a situation, of course, everyone is taking the treatment. How do you establish efficacy of the treatment? You still need to compare to something. So you need some sort of what's usually called an external control of some sort. So now you see that digital twins are kind of a natural answer to this question. Because think about your single arm trial. You have a collection of patients, everyone is taking the treatment. What would you want to know? You would want to know for each patient how they would have progressed over time had they taken standard of care, because that's exactly what you want to compare to establish the efficacy of the treatment. Well, it turns out that that's exactly what digital twins are. We already discussed a digital twin of a particular patient takes information about that particular patient at baseline and gives you a prediction for how that particular patient would have progressed over time under standard of care. So they are precisely giving you the control, what's called the counterfactual, for each observed patient who is taking the treatment. So they're kind of a natural way to construct an external control, which I am calling here, referring to as a synthetic control, just because it comes from a model. So in that sense, I'm referring to it as a synthetic control. Next slide, please. So, so the way this would work operationally is kind of similar. So you have a patient. I apologize here. There should be no randomization. This is a single arm, so there is no randomization. But forget about the randomization box. So everyone goes into the active arm, and in a similar fashion as for the RCT, you have a model that generates the digital twins, which then can be used in the final analysis to compare treated and control patients. Next slide, please. And again, I will not get into the details here, but for people who are interested, there is some more information on this slide. But what I want to stress is that synthetic controls built with digital twins can be integrated with different statistical frameworks that have been developed to do this kind of analysis. And different statistical frameworks provide different levels of robustness in your estimated treatment effect for this single arm analysis. For example, one thing that you might do is just what I just described, do a direct comparison between the observed outcomes of treated patients, which I'm calling Y here in this first direct comparison section. Suppose you have a continuous outcome here, and then you just compare it to the predicted control outcome for each one of the patients in the treated arm, which is their digital twin essentially. And you can just take the difference in means. So you can do that. Of course, now you're entirely relying on the model being unbiased if you estimate the treatment effect in this way, because any bias that comes with M, with the prediction of the model, what that translates into a bias of the estimate of the treatment effect. So there are other methods that have been developed, which I'm reporting here. One example is called they fall into this class of doubly robust method, where you do what I just described, but then you add a correction term that allows you to correct for bias. It involves another model, which is a propensity model. And the advantage is that such an estimator is asymptotically unbiased if either the digital twin model or this propensity model that enters the correction term are well specified, but it suffices that either one of the two is well specified. So without getting into the details, the idea here is that digital twins provide kind of a natural way to construct a synthetic control because they give you the control counterfactual, and then you can use different statistical framework. You can integrate this with different statistical frameworks depending on the choice that you want to make and the level of robustness in the estimated treatment effect that you want to have. Next slide, please. So of course you might ask, why would I want to use synthetic controls? Why can I not use purely data-driven, data-based external controls, such as in propensity score matching? And of course you can do all of this. But one advantage of digital twins is that you're leveraging these machine learning capabilities to learn from large, heterogeneous, often sparse data sets And for example, doing propensity score matching with this kind of data set is hard. So you're kind of leveraging machine learning to handle that part, and then get a natural prediction for the for the control counterfactual via digital twins. And of course, synthetic controls and non-randomized trials can be used, during the sort of earlier phases of the trial, as we mentioned. But what we have referred to as phase 4 and market access post-approval type of questions are often are also rely on randomized trials, and so you can use this kind of technology to answer some of the market access post-approval questions as well. Next slide, please. Okay, I'll go pretty quick on this because I just want to be mindful of time. This is a bit of a more this is still in the analysis bucket, a different application of digital twins. This is a bit more experimental, I would say. We're still kind of fully developing this, but I wanted to share it because I think it's a pretty cool idea. So as we mentioned, digital twins for each given subject, they don't give you just a point prediction for what the outcome of that particular patient will be. They give you an entire distribution under standard of care. So then for each patient, what you could do is that you could compare the observed outcome in an RCT. Let's say you have an observed outcome for a given patient for each patient. You could compare the observed outcome to the predicted distribution under standard of care, which is their digital twin. And you can kind of assess by looking at all the different subjects how likely or unlikely their observed outcome is when I compare it to the control distribution. And I can kind of measure some version of a personalized p-value where what you try to do is you're trying to determine how likely that particular observed outcome is when compared to the control distribution. And you see that by doing that, you are kind of able to understand, you might be able to detect across your entire population responder subgroups, because those will look like as having observed outcomes that are much less consistent with their predicted control distribution. Next slide, please. So so essentially how this idea will work, you would do it in the treatment arm and control arm, you would do some calibration, and then by looking at those p-values, you can use it to detect subgroups. Next slide, please. So the last thing I wanted to discuss briefly is now instead the use of synthetic data. So far, all these applications that we have described, boosting power, reducing sample size, subgroup discovery, single arms, those are all applications of digital twins. There are always subjects actually enrolled in these studies. I'm generating digital twins and I'm using these digital twins in different ways to improve the analysis or help answer certain questions about the treatment. Now we move to a completely different category where we are interested in using this same technology or similar technology to optimize the design of a trial. So the trial hasn't happened yet. There are no real patients. All I can do is that I can use the same generative model. Next slide, please. I can use the same type of generative model that we have described so far, but instead of inputting the particular characteristics of one particular patient, I'm just going to set some constraints of what the population should look like. And those constraints might, for example, be inclusion-exclusion criteria of a particular trial, and then use the fact that this model have learned distributions across baselines and outcomes to sample hypothetical synthetic baselines of subjects that respect these criteria and then predict their progression. So what this looks like is kind of a simulation of what my future trial with those particular criteria might look like. And so as such, this is an example of synthetic data. So these are not actual digital twins of any particular patients, but it is an example of the use of a similar technology, where in this case, I'm using it to simulate a future trial. Next slide, please. And of course, you can kind of see what kind of questions you might be able to answer using the synthetic data. For example, for your future trial, you might want to know, given a set of inclusion criteria, what is the expected progression and variability? How long should the trial be? What happens if I change the inclusion criteria? What's the effect of that on the power of the ability of success of the trial and so on? So there is a long list of questions that you can use this technology to you can use this technology to answer this design questions and try to optimize your future trial. Next slide, please. So here is just a kind of a schematic example. The way this would work is that you define some inclusion criteria, you generate the synthetic baseline cohorts that satisfy those criteria. You predict their progression, then you compute the power. Maybe you want to include some information about how hard it is to enroll patients for those particular criteria to get some combined measure of probability of success. And then you might want to, once you have this machinery, you can explore the effect of changing some of these inclusion criteria. Next slide, please. Yeah, and here's just a summary of the advantages of this kind of approach, which we've already discussed. Next slide, please. So the last thing I want to mention here before I get to my summary is that those are examples of applications of AI and digital twins, of AI, digital twins, and synthetic data. Whenever these applications need to be put in front of regulators, then I wanted to remind everyone that there is a FDA draft guidance that came out in January 2025 that describes 7 steps. It's a risk-based framework that goes through 7 steps, and each one of these applications or these examples, if that application had to go in front of regulators, can be reframed in terms of this 7 steps. But even if it doesn't have to go in front of regulators, I still find, the 7 steps, in particular, the definition of the context of use, assessing the model risk, and developing a plan to establish credibility. I find these steps very useful, and they provide a framework to think about, actually using these technologies in actual trials, even if they're not necessarily going in front of regulators. It might just be for internal decision making. Last slide is a brief summary of what we've discussed today. So we've discussed different applications that go from of digital twins that go from reducing sample size to boosting power in a trial, how you can use digital twins as synthetic controls, how you could potentially use them to find responder subgroups, and lastly, how you can use synthetic data to simulate and optimize trial design. Of course, those are just a few examples. And as I just mentioned, when we think about these applications, it's always important to frame them within, think about the context of use, risk assessment, and risk mitigation. That's all. So thank you so much for your time.
Thank you so much. This was just terrific. I think we can take down the slides because we have a number of questions. I'm sorry, I'm hoarse, that have come in. But let me start with one. When you defined your digital twin in the beginning, you said it was predicted likely progression under the placebo. But then when you were talking about it, you talked about standard of care. So how do you resolve that? And when you think about standard of care, what if that standard of care changes? Do you now have to do a different adjustment?
Yeah, that's a good question. I should have been more precise. So if the digital twins are used in an RCT, like in the Procoa application that we described as covariates, then you do want those to be a prediction of the outcomes under placebo. So, for example, if there is a placebo effect in the trial, you want the digital twins to model that. The
In
way,
both
yes,
arms. Yeah.
yes. So the way you do that is by essentially making sure that the model is trained on past clinical trial control arms of past clinical trials effects as in your target trial. Now, so that's one consideration. As for the sort of, as you say, underlying potential shifts and changes in standard of care, that's also a question that goes back to the training data. So if, in fact, there is a change or shift in the standard of care, you want to make sure that your training data as a representation of that and your model is able to capture that. One last thing I will say is that for the particular application of Procoa, where you're using digital twins as covariates to improve the efficiency of the trial, there is one particular metric that matters, and only one metric, which is how your predicted outcomes, so your covariates, how much they are correlated with the actual observed outcomes. And what that means is that in principle, even if there is a change in standard of care and that change was just, let's say, in the simplest case possible, was just a constant shift for everyone, that would not impact, in principle, the
Right.
correlation. So you will still get back the performance that you wanted. That said, you still want to, of course, capture as
Be
much as you
aware of it.
can.
Yeah.
Yes.
And if you're doing a single arm trial, my assumption is that it would have to be against standard of care as the natural evolution. Is that true or not true?
Well, I believe if that depends. I believe that if you think that even in the single arm case, there is a placebo effect, which I believe in some cases there can be,
There
then
is.
you would still want to model the placebo effect for your external comparator as well.
And has EMA or FDA or other regulatory authorities validated Bayesian Procoa? And do you expect any challenges with that? If not,
So the Bayesian version of Procoa, as I mentioned, because it does not guarantee strict control of
type
type 1
one.
error, likely it cannot be used for a phase 3 trial, but it can be used in earlier phases. Um, I am not sure whether we had interactions with FDA on the particular Bayesian Procoa application, so I cannot say for sure that we have had interactions. But I would expect to be no different, the type of interactions to be no different than the other Bayesian analysis, because it is effectively, essentially, a form of Bayesian analysis. In fact, it's a combination of the
Yeah.
covariate adjusted, which is already, sort of within the guidelines. It's just a Bayesian version of it. So whatever conversation you might have with a regulator around the Bayesian analysis, those same, kind of constraints would apply to the Bayesian Procoa case.
So I have one other question because we only have 1 minute, but there are many in the chat and we're Q&A and we'll get to them a little later. Maybe not today. So in your analysis of Procoa and Bayesian Procoa, you applied all of the power to the new treatment arm. Is there any ever reason, let's say, if the new treatment, the hypothesized treatment, is potentially more toxic, but much better, that you might apply your gain to the control arm and decrease the treatment arm?
Yeah, you can do either. So there are all sorts of things you could do. So you could,
Yeah,
you can think of having some sort of credit in efficiency. So you could use it to reduce just the control arm, to reduce just the treatment arm, to reduce both of them, to spend some of that to reduce sample size and some other to boost power, to
yeah.
spend it all these combinations are possible depending on the specific use case. The reason I mentioned reduction of control because typically, people don't want to get placebo, and so that's a kind of a natural application where you
Yeah.
have fewer people taking placebo. But you could imagine different scenarios for sure.
So thank you so much. There are questions in the chat. I think what we're going to do is probably come back to those at another occasion and continue to explore, maybe with a panel, some of the questions that have been raised in the applications. But once again, thank you so much for a really brilliant explanation of the various uses that we should be considering with a sophistication, yet a simplicity that even I thought I understood. I'm sure I didn't, but I had that moment where I thought I'm following him. So thank you so much. And to the audience, thank you for joining us. Happy Thanksgiving. We'll see you hopefully in December or January. Thank
Thank
you.
you so much.
Probably January. Thanks.